Okay, in this session we're going to specifically look at apoptotic signaling, and this is the extrinsic apoptotic pathway, and we're going to focus on death receptors. So over the course of this session and the session next week, we're going to cover both intrinsic and extrinsic apoptosis. This particular video this week is going to focus on extrinsic apoptosis, which is death receptor mediated apoptosis. So by the end of this week and next week, you should be able to do the following. So describe the death receptor mediated apoptotic signaling pathway and also the intrinsic mitochondrial pathway, which I'm going to cover next week, and importantly show how these two pathways are interrelated. I'm going to talk mostly about death receptor mediated apoptosis today, but we're going to touch on aspects of the intrinsic pathway, and then next week we're going to focus in detail also about how this intrinsic pathway is regulated by P53. So that's what we're going to cover over this week and also in next week's session. And in the course of these next 2 weeks, we're going to cover what looks like an incredibly complicated diagram, and this is the apoptotic signaling diagram, which covers all of the key features of programmed cell death apoptosis. Now I'm going to break this down, and I've color coded it so that you can make more sense of it, but basically what it shows is that these here are death receptors, and we're going to cover death receptors in this session. So FAS and tumor necrosis factor receptor, and also death receptor 4 and 5, which I'll cover in this session as well. And they are the receptors, and they can be bound by a ligand, and this ligand is on the surface of something like an immune cell, like a cytotoxic T lymphocyte. So FAS ligand is on cytotoxic T lymphocytes. TNF can be on the surface of many other cells. Trail, I will talk about shortly, on the surface of monocytes and natural killer cells. So a death ligand in red can bind to a death receptor and then pass on a signal to an enzyme called caspase, and in yellow is the initiator caspase, caspase 8 and 10, and then they pass on the signal to the executioner caspases, caspase 3, 6 and 7, and these executioner caspases degrade the cell. So where we've just seen that the cell gets degraded partly by cutting up DNA, and we get DNA fragmentation, that is a direct result of usually caspase 3 cleaving something to allow a caspase activated DNase to kill the cell. So it looks complicated, but for now you need to understand death ligand in red, death receptor in green, initiator caspase in yellow, and executioner caspase in blue. Now that is extrinsic apoptosis because that comes from signaling outside the cell, typically a ligand on an immune cell binding to a cell and initiating cell death by activating the death receptor. There is another way of killing the cell, and that's what we call the intrinsic apoptotic pathway, and the intrinsic apoptotic pathway works via the mitochondria, and that intrinsic signal could be something generic like hypoxia, low levels of oxygen, it could be DNA damage, it could be generalized cell stress, and that is picked up by the mitochondria, and we'll find out how next week. The mitochondria will release its own initiator caspase into the cytoplasm, and that initiator caspase, called caspase 9, will activate executioner caspases, and those executioner caspases will degrade the cell. So two different signals, one from outside the cell, one from inside the cell, so we have extrinsic and intrinsic apoptosis. And this can be summarized here. You've got the extrinsic pathway, you have death receptor activation by something like a T cell or a natural killer cell, causing caspase activation, causing executioner caspase activation, and then death. The intrinsic pathway, something like hypoxia, DNA damage, you get both cytochrome C release, which I've not yet told you about, and caspase 9 release from the mitochondria. That results in caspase activation, and then that results in death via executioner caspases. And we're going to look at all of these pathways, both the receptor complex here, the death receptor complex, the different forms of caspases, and when we get onto the intrinsic pathway, we're going to look at cytochrome C release from the mitochondria and how we get release of things from the mitochondria, but that will be mostly next week. So this summarises the two different pathways. The extracellular pathway or extrinsic pathway is cell signalling from death receptors on the cell surface being bound by a death ligand on a different cell, whereas intracellular intrinsic pathway, the cell is sensing something going on within the cell, such as double-strand DNA breaks, p53 pathway getting activated in response to DNA damage. UV radiation causes DNA damage, that will cause intrinsic apoptosis. So will withdrawal of growth factors, and so will reduction in oxygen, what we call hypoxia. But also the activated extrinsic pathway will also cause the intrinsic pathway to become activated as well, and often extrinsic apoptosis works through intrinsic apoptosis. And I'll just draw your attention to this little arrow here going from active caspase 8 heading towards the mitochondria. That is the link between the two pathways. This protein called BID, when caspase gets activated, and that only gets activated when the death ligands bind the death receptors, BID can become activated. That can travel to the mitochondria and kickstart an intrinsic apoptotic signal as well as an extrinsic. So the extrinsic signal can either go direct, so death ligand, death receptor, and go direct to the execution of caspases, or it can also pass on signals around this route and boost the signal. So first we're going to concentrate on the death receptor complex. So this is the diagram that you've just seen, but in a bit more detail. Here's a lymphocyte expressing Fas ligand. That is a death ligand, and other death ligands exist. So death receptor 4 and death receptor 5, those are receptors which will bind the death ligand trail, the TNF related apoptosis inducing ligand. Fas is the receptor for Fas ligand, and TNF receptor 1 is the receptor for tumour necrosis factor alpha. Now these death ligands are trimeric, that is 3 proteins clustered together, and their job is to cluster together 3 receptors in close proximity. And we'll call this a homotrimer because it's 3 of the same type of death receptor. Now as that happens outside the cell, that links together 3 intracellular domains of the death receptor, and those 3 intracellular domains of the death receptor, when they come into close proximity, recruit another protein, and that protein is called FAD, the Fas associated death domain protein. And FAD has one key job, and that key job is to recruit a protein called procaspase 8. And procaspase 8 is a proenzyme, so it needs to be cleaved to be activated. FAD has got 2 ends. One end is a death domain, and the death domain of FAD sticks to the death domains here, these trimerized death domains. The other end is a death effector domain, and that death effector domain will bind to the death effector domain of procaspase 8 or procaspase 10, and that will bring 2 caspase 8s in close proximity to each other, and then they can auto-activate each other, and we'll see this in more detail in a moment. Once this has occurred, so ligand binds receptor, trimerize the receptor, bring in FAD, and then bring in caspase 8, we call that the death-inducing signaling complex, and that signals an apoptotic signal to the cell. So this is just shown with some slightly nicer diagrams. Here we can see a killer lymphocyte, so a cytotoxic T lymphocyte expressing Fas ligand on its cell surface, which is a trimer. Here's a target cell, which for whatever reason needs to be executed. It expresses Fas death receptors on its cell surface. The T lymphocyte binds the receptor, that trimerizes the receptor. Once trimerized, that brings in FAD, the Fas associated death domain protein. That will bind into the death domains. The other end is a death effector domain, and that death effector domain will bind to a death effector domain on procaspase 8. We get multiple caspase 8s brought in close proximity to each other. They can auto-catalyze each other and activate each other to create active caspase 8 protein. And that active caspase 8 protein kickstarts the apoptotic process because that active enzyme will activate the executioner Caspases. Now, because the result of ligand binding to receptor is certain cell death, this is very tightly controlled. So cells have ways of bypassing or resisting T cell and natural killer cell mediated apoptosis, and they can do this in a number of ways. One of the ways is to express decoy receptors. Now, trail, which is on the surface of our monocytes, and some of our natural killer cells, binds to death receptor 4 or 5, and that creates a DR4 or DR5 homotrimer, and that induces apoptosis. Now, normal cells also express decoy receptors which can bind to trail. We call these decoy receptor 1 and decoy receptor 2, and these decoy receptors can bind trail in a trimeric way, creating a heterotrimer, and therefore this doesn't signal apoptosis because there's only one death domain here. If there's if there's only one or two death domains, then it doesn't signal apoptosis. So that's one way. The presence of decoy receptors can prevent apoptosis, and also soluble decoy receptors. So a lot of the research that I did quite a few years ago was to look at this role called OPG, or osteoprotegerin, that is secreted from cells, and that can bind to trail and prevent it from getting anywhere near the receptors. So if a cell is producing high levels of OPG, any T cells that come, any natural killer cells or monocytes that come near it will have their trail bound up with OPG, and that will stop them from killing the target cell. So there are various ways of which normal cells can avoid apoptosis. So we're going to move on to the caspases themselves, and these are caspases are cystine-catalyzed aspartate targeting proteases. That's where the name caspase comes from. They are mostly, I would say mostly, quite a lot of the caspase enzymes induce apoptosis. There are other caspases that do other things, but a whole large family of them induce apoptosis. And they all have the same generalized structure. They have an N-terminus, they have a C-terminus. The N-terminus is crucial for binding into the disc, the death-inducing signaling complex, and then they have two cleavage sites. They are cleaved away from the death-inducing signaling complex, and they are also cleaved in the middle to produce two heterodimers which come together to create one functional enzyme. So by cleavage here and here you get one of these, and this is one full active caspase subunit, and all of the caspases operate in the same way. Now at the death-inducing signaling complex, these are brought close enough to each other so that they can auto-activate each other, and that's why the trimeric death receptor needs to bring in multiple caspase 8s to kickstart the process. Now these caspases recognize a very small number of proteins, one of which is itself. The other proteins it recognizes are executioner caspases because it cleaves them in the same sort of way to create active executioner caspases. And I mentioned the protein BID earlier on, the link between extrinsic and intrinsic apoptosis. Caspase 8 will cleave BID into a truncated form, and that truncated form will go to the mitochondria and kickstart apoptosis at the mitochondria. So this can be summarized here. At the death-inducing signaling complex, you get activation of one initiator caspase. That can activate many more executioner caspases, and executioner caspases will be floating around in the cytosol in the inactive form. They need their N-terminus cleaving off, and they need to be separated into their large and small domains, and they need to come together as two heterodimers. Once caspase 8 has been activated, it can activate the executioner caspases, let's say caspase 3, 6 and 7, and they can then cleave their targets, and those targets could be things like nuclear lamin, which I'll talk about shortly, and other cytosolic targets to cause a cell to become degraded. Now, as I've alluded to, the intrinsic pathway has caspase 9. Caspase 9 is an initiator caspase just like caspase 8. So once a mitochondria gets activated, caspase 9 is released, and that becomes an initiator caspase which then acts on executioner caspases in the same way that caspase 8 and 10 does this. So I'm now going to talk about what these executioner caspases do once they become activated, and one of the key proteins that you need to know about is ICAD. Now ICAD means the inhibitor of caspase activated DNAases, and caspase activated DNAase is called CAD. So ICAD binds to CAD and prevents it from being active in a normal cell. Once apoptotic signaling has started, the ICAD gets degraded, releasing the CAD, the caspase activated DNAase, and that is then in the nucleus, and it will degrade DNA by cleaving between those histone proteins, giving that classic DNA laddering that we will see in next week's lecture on apoptotic detection methods. But you'll have to believe me for now, you get fragmented DNA that can easily be seen in multiples of 180 base pairs. In addition, caspase 6 becomes activated, and that will cleave lamin, and that will cause the nuclear membrane to change its structure and to allow chromatin to collapse, but also allow the nucleus to be broken down into smaller fragments for phagocytosis to occur. So nuclear lamin is broken down to allow the cell to be broken up into several pieces. In addition, active executioner caspases will cleave a protein called gelsolin, which in itself naturally cleaves actin filaments in a tightly regulated manner, but when caspase has cleaved gelsolin, it's in a constitutively active form, which will then go and fragment all of the actin cytoskeleton. And the actin cytoskeleton is like an internal scaffold within the cell, and the cell cannot be broken down into apoptotic bodies whilst that cytoskeleton is intact. So activated gelsolin by cleavage with a caspase allows the actin cytoskeleton to be degraded. And finally, we get cleavage of PARP. PARP is poly-ADP ribose polymerase, and this is part of the DNA repair program. So if PARP detects DNA damage, that will activate a DNA repair process, but we want the DNA to be fragmented and not repaired. So caspase 3 will cleave PARP into its inactive form, and that will prevent it from flagging up the caspase activated DNAase damage to the DNA repair system. We don't want that to happen. So we're effectively disabling DNA repair with caspase 3 so that the DNA can be fragmented. What we don't want is a cell that's programmed to undergo apoptosis to repair all of its DNA, shove all of this DNA back together, and then survive that pro-apoptotic signaling. So this is a nice diagram of death receptor signaling. It just happens to be trail. Trail binds to death receptor 4 and 5. Trail can bind to decoy receptor 1 or decoy receptor 2. They don't signal apoptosis, but if trail binds to death receptor 4 and 5, that will signal apoptosis. We then get FAD recruitment. FAD is a double-ended molecule, a protein, and one end will recruit procaspase 8. Procaspase 8 will then auto-activate itself to produce active caspase 8, which will produce active caspase 3, which will cause the cell to undergo apoptosis. Active caspase 8 will also cleave BID into TBID, truncated BID, and as we'll find out in the next session, that will translocate to the mitochondria and encourage both cytochrome C and caspase 9 to come out of the mitochondria, and I'll cover this in more detail next time. That will form active caspase 9, that itself will activate caspase 3, and that will drive apoptosis. And these are just some extra control mechanisms that are in place to regulate the process. Now this can be summarized in this little diagram, and this is from an article that I wrote on how the apoptotic pathway can be manipulated by drugs to promote cell death as an anti-cancer therapy. Now one of the things that I work on is the protein trail and how trail signaling can drive cancer-specific apoptotic cell death. And the sorts of experiments that I do is add trail to cells and try to enhance its signaling with different classes of drugs. And all of these drugs down here and all of these drugs down here are all known to enhance apoptosis, and what my research has shown is since this was published, we've got a few other classes of drugs to add to this list that can basically stress the cell, usually to enhance the intrinsic apoptotic pathway, and therefore an extrinsic apoptotic signal becomes more potent, and that's where much of my research is focused at the moment. So that is the end of the extrinsic apoptotic signaling pathway, with just a hint of the intrinsic pathway as well. You've got a glossary of terms there of the things that I've talked about, and that should help you when you go back and look over the notes. And then have a little think about this question: how can apoptotic changes be analysed in the lab? So what is it that's different about apoptotic cells compared to normal cells? Have a think about how you could do a biochemical test in the lab to assay for either specific apoptotic related proteins or effects that have happened. I've shown you a few tests. I've mentioned DNA fragmentation, and that can be related to the nuclear morphology of the DAPI staining. But have a think about anything else that you could assay for that would be specifically active in an apoptotic cell compared to a non-apoptotic cell. And we'll talk about possible apoptotic tests in the live session. Okay, so that's the end of extrinsic apoptotic signaling.